Hello, my name is Chang. I'm from Alice Ting's lab at Stanford. So my project is developing next generation proximity labeling enzymes, which is called FlexID2 and LaccID2, which we thought it will has like more advantages over the current proximity labeling enzyme. So proximity labeling is proteomic technology that help you to profile protein-protein interactomes and compartmentalize the proteomes in living cells and organisms with very high spatial and temporal resolution. So basically, we will fuse the PL enzyme to the target protein, then it will generate like small molecule active species. It can label the surrounding proteins. Previously, our lab already developed like two widely used enzymes called APEX and TurboID, which use like biotin-based substrates and generate like biotin-based active species and label the surrounding proteins. Then with the biotin tag, we can pull down the surrounding protein partners and then do mass spectrum to identify those. And however, we think these two enzymes are not perfect yet. So for example, APEX2, which needs hydrogen peroxide, is supposed to be like super cytotoxic. And TurboID can only use biotin, which means like it can be affected by the endogenous biotin-related background. So we developed these two new enzymes called FlexID2 and LaccID2, which is supposed to solve these problems. So for example, the LaccID2, it's a multi-copper lacase that can only use oxygen as an oxidant to generate a biotin substrate, and then it can label surrounding proteins. So we think it's more gentle than APEX, and it has like super high selective surface activity, so that it's very perfect for the surface proteome profiling. And now we are using like continuous yeast display as the strategy to evolve this enzyme to become more resistant to endogenous inhibitors such as serum, diones, halides, and the pH. And in the end, hopefully we can apply this to the in vivo system. And the other enzyme that I'm very excited about is FlexID2, which is come from E. coli lipoic acid ligase, and we engineered the pocket to accept various of substrates such as azide, alkyne, and other clickable handles. And we found out that there's a very interesting feature of this enzyme is that like the off rate of this enzyme is very slow. That means the substrate is kind of retained in the enzyme pocket until the other proteins like come and attach to this protein. Then it can transfer the substrate onto that protein interactor. So we think it's kind of unique contact-dependent labeling, and we think it can provide you with super high specificity. So now we are applying a very aggressive engineering method called mammalian cell-based directed evolution to evolve this enzyme to become more active and more stabilized. So if you are interested in like spatial proteomic and enzyme engineering, directed evolution, please come and visit at poster number 22, and we can chat more. Thank you so much.